This is part 2 of a 3-part series, Your Life is Not an Accident. Where we left off in part 1, we were talking about living cells, and all living things are made of complex cells, from a simple, not a simple, but from a bacteria 1 cell to humans. Inside every cell is information that determines how that living organism is built, whether a plant or an animal. And there are no changes, there's no evolution or changes or variation to living things unless there's first a change in its information. Now, a computer software that we use every day in our computers, our desktops, and our mobile phones are made up of a 2-digit code. It's either a 1 or a 0. The software in all living things is somewhat similar, but a little more complicated. It's a 4-digit code, A, T, C, and G. These are nitrogen bases, adenine, thymine, cytosine, and guanine, but I'm just going to use the letters. Now, the digital code is read, the coding that builds structures is read vertically, up and down, kind of vertically if we were to take that DNA strand. You see the various letters there. Now, on the left is the coding strand, and that is where the information is made. There is no chemical attraction. There's no bonding that is going on. The only bonding that occurs is on the opposite side. And the A's always bond with the T's. Apples go on trees. And the G's and the C's always bond together. Cars go in garages. And so the point is, whenever that cell duplicates, because of that bonding that occurs on its complementary side, the other side, it'll always attract and make an exact duplicate of the DNA whenever the cell divides. Now, all of those black lines are kind of like chemical attractions or bonding. You notice that there's no chemical attraction or bonding here. And if there was, then the information would be repetitive. Every time you got a G, you got a T, and it would be GT, 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 GT. But information is irregular and non-repeating patterns. Now, the DNA is found in the nucleus of every cell in your body and every cell in other animals. The DNA is the most incredible information storage system in the world. The amount of information that could be stored within DNA, the size of the head of a pin, would be the equivalent of paperback books stacked 500 times further than the moon from the earth. That is quite an incredible storage system. All living things are built by DNA. Another way of thinking of DNA, it's a type of assembly manual. It makes people, birds, plants, trees, insects. Now, the size of the amount of information in the DNA of a bacteria would fill about 2 books of 500 pages each. For a human, that would be about 2,000 books. So one of the things that natural process has to explain, if evolution is true, is how does information increase by 1,000%? What is the mechanism for that? Now, just to do some terms here, and don't worry about it, this is just kind of a big global picture. Think of the letters A, T, C, or G. They're called nucleotides, and they're equal to, like in a book, a letter. Then we have what's known as the 3 letters together, 3 nucleotides, 3 letters, forms a word. And that'll eventually code for amino acid. We'll explain that later. Okay, so 3 letters equal a word. Then we have genes, and think of genes as like books in a library. And in a human, you get 1 copy of your genes from your mother and the other from your father. Genes are a section of the DNA, and they make a protein, and we'll get into proteins later. And genes range in size from 200 letters of those individuals, ATs, Gs, Cs, or up to 2 million letters in a protein. Next, we have chromosomes, and in a human, we have 46 chromosomes. Think of them as like shelves, 46 shelves in this library. The chromosome helps package the DNA into smaller volumes so it can fit inside the cell, and it also helps prevent DNA damage, which is very important. And finally, we have what's known as the genome. That's the entire library of all shelves, of all books. That's what builds you. Now, this information, it is linear like a book. In other words, it's read A through Z. But it has countless loops like a computer program. It has genes that regulate other genes. It's self-regulating. It's multidimensional. There's higher levels of information. And by multidimensional is there are codes within a codes. There's no human information system that can even come close to comparing to it. Now, we think of bacteria as very small and simple. It is not. Bacteria is built by DNA. And a bacteria has on a rotary motor, it's the flagellum that turns around and helps propel that bacteria. The genes determine the parts in assembling that rotary little motor and flagellum. In fact, if we go into the cell membrane, we see in that flagellum that whips around, there's O-rings, bushings, drive shafts, U-joints, rotor. That's composed of 40 parts. All the parts have to be there at the same time, or else this would not function. And incredibly, what causes that to spin around, that flagellum, is an ion-driven little electrical turbine motor. And it can whip that around at 17,000 revolutions per minute, and it can reverse itself in half a minute. Incredible, incredible design in something as small as a single-cell bacteria. Now, a sequoia seed that builds the large trees, its entire DNA is within the seed. And what activates it is water and nutrients in the soil. And when that occurs, it starts to germinate. The DNA information grows roots. It builds photosynthesis machinery, builds green leaves, and builds pine cones with millions of new seeds. So from the genome, the DNA in the sequoia seed, it builds these tall, gigantic, beautiful trees. Now, your one cell beginning came from the egg no bigger than a period at the end of a sentence. Half of it came from your mother and the other half came from the father. The egg from the female is the largest cell in the body, and the smallest cell is the sperm. Now, when the single cell, when the egg is united with the sperm, that egg turns into two cells. And then it turns into 2, turn into 4, and 4 into 8 and 16. By day 3, there are 16 cells. By 2 months, all the basic parts driven by the DNA instructions have been built. The heart has been beating after 1 month, and so the parts just need 7 months to mature and develop. Now, when that one cell makes 16 and 32 and 64, at some point in that 2-month process, it starts making different cells. You have over 210 different cells in the body. The nerve cell, the liver cell, the bone cell, skin cells, red blood cells are all different. Muscle cells, fat cells, connective tissue cells. And what the DNA starts to do is eventually those 210 cells are driven to an endpoint to create a functional tissue, let's say a muscle. And those tissues form organs like a heart. And then those organs are part of another system, like your circulatory system, consisting of veins and arteries. In the human body, there are 10 body systems. When the embryo of the human is being developed, within 2 months, most of all of these systems are built. The digestive system, the endocrine system, the lymphatic system with white blood cells, muscular system with 650 muscles and tendons, and nervous system, brain, spinal cord, reproductive system, skeletal system of 206 bones, respiratory system of breathing and bronchial tubes, and the urinary system. Now what's incredible is all of these systems are dependent upon each other. What's the point of having a heart and a circulation system if there's not a respiratory system to circulate the oxygen? If one of these doesn't function, there's death or extinction. So the question is, how does one entire library in every cell builds different types of cells? The New York Times in the Science edition said this is a mystery, is that a variety of these specialized cells collaborate into building a body of different cells, yet they have the identical genome. I think this is probably the best way of saying it. Like actors with the entire script, certain cells can only see and read their assigned parts. So each cell only expresses or turns on a fraction of its genes. It's kind of like on-off switches. And this is known as gene regulation, and as the scientists have said, this critical process is still not fully understood. So the question is, how does the DNA do it? We still haven't explained that. How does it build all of these different structures in life of different organisms and different levels of complexity? Well, I'm going to talk about the size of your library, the genome. The nucleotide is one letter, okay? And typically when we talk about the size of the entire library, we talk about base pairs. So there's, the base pairs is it's complementary. So base pairs is half the amount of letters of nucleotide. So let's just get into that. This is showing the size in books at the top and in millions or billions of base pairs. The red line in the middle is the amount needed to build a human being. If we look on the far left, that's what we need for bacteria, from the smallest, or less than a book, to a little bit bigger. The bacteria are single cells, and they're 10 times smaller than plant or animal cells. And you have a lot of these in your body. They're important for your digestive system. Now next we have plants. Look how large they are, and fish. And they have the largest library or genome size. In fact, the largest known size is this flowering plant. It's got 149 billion, whereas a human has around 3 billion. Now, you don't want necessarily a very large genome, because whenever the cell copies itself, it's got to copy this huge library of information. And these plants are very slow growing, and they are also respond very slowly to environmental changes. Next, we have amphibians, reptiles, birds, and finally mammals. So you can see that there is a minimum amount in each category to have, but the size does not necessarily represent the complexity. A lot of these are hovering around 5,000 books, and if you get up to the far end of the plant, we're talking about 500,000 books. So when the cell divides, it's got to duplicate its entire library before dividing into 2 new cells. And the reasons for dividing are about 3 major reasons. One is to grow. As you grow, cells don't get fatter. As you grow and that embryo gets bigger and bigger and you grow as an adult, more cells are added. The second is to replace damaged cells. If you skin your knees and there needs to be repair, then it makes more cells to replace those damaged cells. And third is to replace worn out or dying cells. Cells don't have the lifetime of your body. In fact, colon cells only last 4 days, skin cells 3 weeks, your red blood cells are 4 months. You get a new stomach lining of cells every 3.5 days, but your brain cells do last an entire lifetime. You don't want those changing because your memory and information are stored there. So when the library, the genome of the DNA is copied before it's divided, there's the original cell there. It unwinds and starts making 2 new strands before it divides. And here is some of the molecular machines. They're protein enzymes. There's one on the left that holds it in place to keep the original from unwinding, another one that winds it, and these 2 new strands are being built nucleotide by nucleotide and an incredible speed with these kind of building machines, if you will. Now, whenever it duplicates, there's a quality control. And Science, which is one of the premier peer-reviewed journal in America, said this: when copying errors are made, the cell has machinery that identifies and repairs to maintain the organism's genetic code. Very important because you don't want errors to come in. Although not error-free, genes not repaired or bypass. This is kind of important because evolution relies on copying errors to build greater and more complex. But Science magazine says there's a repairing system here. Now, what are we talking about? The implication of this is the enzyme machinery will check the new daughter DNA strand and to fix or repair it to make sure it is identical to the original. There's an intelligence there because you have to distinguish the original from the copy because you don't want to make changes to the original. The enzyme must therefore know what is correct to make the repair. Incredible layer there. Now the DNA must duplicates its entire genome as cells need to be replaced. But it also, in order to build the segments, must make copies of small segments of the DNA to build proteins. And that's what's known as our genes. And all of our traits come from these genes. We're going to get into proteins pretty soon. You are made of protein. I mean, they are the large, complex molecules. There's over 100,000 different kinds in the body. Muscles, hair, skin, eyes, body organs, digestion, circulation, the communication within cells, enzymes, motions, the machinery inside your cells. People ask, what about carbohydrates and fats? Well, they're created by proteins. They're not as complex as protein, and mostly they're used for energy and storage. Once a protein is formed, it can exist for minutes or years. The average in a mammal is 1 to 2 days. So these proteins need to be replaced. They degrade, and when they degrade, they are recycled inside the cell's machinery. And you got to have that level's got to stay within a specific range for that cell to function properly. Now, proteins, this is incredible, are made up of amino acids. And if you look at a protein shake, you'll see a lot of those amino acids listed. For the protein in the body, there are 20 different amino acids. And they're long chains put together, and those 20 different ones have to be in the right order. The average size of a protein is 466 amino acids in a specific order. There are 17 in this slide here, little dots representing amino acids. What are the odds of just getting 17 right in the right order? The odds are 1 in 355 trillion. So the odds of getting 466 amino acids in the right order is beyond imagination. It's a number so large, it's larger than the number of atoms in the universe. The smallest protein is 10 amino acids. It's found in the saliva of a Gila monster. And the largest protein is 30,000 amino acids. It's titin. It's found in our body. It allows the elasticity in the muscle tissue. So there are long chains of amino acids, and then to have function, that long string of amino acids is folded into a protein in order to have function. The precise arrangement of amino acids determines its shape and its function. You change the amino acid and you change its shape and function. Any change will result in a loss of its shape and function. Okay. Now, in a computer, they have 8 bits, and 8 bits, that's either a 1 or a 0, will equal a byte. Let's say it's a character. So that is the, if there's 8 bits, how many maximum number of bytes can you have? Just hang in with me. If it's 2 times 2 times 2 times 2 times 2, it's 2 times itself 8 times. Because we have an option of a 0 and 1 in the first place, a 0 and a 1 in the second place. You come up with that of 256 characters. That's enough for the 26 letters in the alphabet, upper and lower case, numbers, special characters. In some computers, they've gone to a 16-bit string, and that's 2 times 16 and that allows for characters from the ancient Greeks to all the modern language. But let's just stay with our 8 bits. So when we in the computer want to create the word hello, we have to say where the word begins. So 6 zeros and a 10 is a start. And then at the bottom, 6 zeros and 11 is a stop. And that's the actual code for an uppercase H and a lowercase hello. And that spells out hello. In DNA, they use a 3-letter code, and that determines what amino acid is used. So AAG equals lysine, CGC is alanine, and CAT is the amino acid histidine. And so whenever the DNA is coded, is read, it sees these in groups of 3. Because the 3 letters are like the words that determine the amino acid. Now, in a protein, it's got to know, out of an average of 466 amino acids, where to start and where to stop. Where does this protein begin and end? And just like in the computer, ATG tells it this is the start of this protein or gene, and at the bottom, TAG says this is the end. And in between, you're going to have various letters, up to 466 of these different words to form a protein. Now, the coding strand, whenever a DNA is built, whenever a protein is built, it's not built inside the nucleus where the DNA is stored. It makes a copy of itself, and then that copy leaves and it builds it outside the cell. So it doesn't use the coding strand to make a copy of itself. It uses the template strand. And by using the template strand, if you look down, you get an exact duplicate of the DNA, because that is the coding strand that determines the amino acid. So that will leave the cell, and it goes into a ribosome. And the ribosome is a factory, and I'm going to show you a video of this because you got to see it to believe it. The factory is able to read the instructions on the copy of this gene here. And then this is inside the ribosome. What happens is the upper level is that long segment of, it's called a messenger RNA. It's a portion of your DNA library. And then these little transfer RNAs, the amino acids are on the bottom, and they start building an amino acid. So We've got to see that, and I'm going to show you an animated video of how that happens and narrate it. When the mother's DNA and the father's DNA unite, it starts a process of life. And within a very short time, the DNA builds different cells, 10 different systems that are all interdependent. And within 9 months, a living baby is born. Now, as we go inside the cell to see what kind of activity happens, we have the energy plants, mitochondria, and there's tubules that lengthen and shorten, help control traffic. And here's like a UPS machine. Instead of driving, he's walking a very heavy molecule. He knows where he's going because there are molecular tags that tell these various carriers where to take these molecules in the cell. Now we're going inside the cell's nucleus. There's actually over 2,000 to 3,000 books of information. Here's a molecular machine is unwinding it because we're going to make a copy called messenger RNA. It's a strand of instructions on how to make a protein, how to build it. This is the nuclear pore that regulates traffic in and out of the nucleus. And now this strand of information, coded information, is going into a factory called a ribosome. And their transfer RNAs will bring in amino acids. Notice the 3 prongs. Those help define what an amino acid is. And then it goes into a machine called a chaperone, in which it helps assist fold the protein into a usable shape. And then after it is completed, another type of protein carrier takes it to where it is supposed to go. That was a pretty amazing video. And just imagine, we're supposed to believe that all of those machines and all of that complexity just happen by accident. The question, as we saw that long amino chain that was folded into a protein, how does that take place? Well, some long chains will fold spontaneously based upon the precise order encoded by their DNA. Others need help, and a cell has an army of assistants that monitor, coax, and repair these long chains into folded proteins. They're called chaperones, kind of like an adult chaperoning a high school prom. These are various examples of protein chaperones that help fold amino acids into a usable shape. The question is, how does one protein, these chaperones, know how much information a completely different protein needs to fold into these 3-dimensional shapes and structures? And if a protein has to be there to build other proteins, how did the first protein originate? Now, they used to think that proteins kind of binded together like logs on top of a fireplace, just kind of laying on top of each other. But the shape and the chemical bond is like a lock and key. There's a specific shape that has to be there for that bonding to take place, making it a little bit more difficult for this to happen by chance. Now here are examples of some proteins folded into shape. These were once long chains of amino acid, and the cells produce over 100,000 different types of proteins. Here's some of the examples. Quite beautiful. Nice job of folding. Here's some others. The one on the right is an antibody protein that's in the human's immune system. Now, in 2005, IBM built what's known as the Big Blue. It's the most powerful supercomputer ever built. And according to their website, the reason why? To help biologists observe the invisible process of protein folding. According to their website, it'll take 1 year for the supercomputer to finish calculations to model folding of a simple protein. The cell takes less than a second. Well, that concludes part 2. I hope you stay tuned and see what I consider the most important, and that is part 3, We're going to look at what is evolution's mechanism for evolving creatures and making them more complex and why it won't work. And I'm also going to show you from actual textbooks deliberate misrepresentations of facts about evolution. Thank you.